Okay, so we are beginning now talking about DNA replication, and we're going to begin first by talking about how prokaryotes do the replication, because a lot of the replication machinery were discovered by studying bacteria, especially E. coli. And so we'll talk about the terms that are associated with replication listed here, and we'll talk about them with respect to the prokaryotes, and then also, of course, with the eukaryotes after. And we'll describe replication in prokaryotes and the purpose of our RNA primers and things of that sort. Now, in prokaryotes and eukaryotes, replication is called semi-conservative. And it's semi-conservative because each new double strand have one parent and one new strand. And then so having the parent strand is important because you want to make sure to maintain fidelity. So fidelity is going to require fixing mismatched bases, and it's always going to be the parent strand that is going to donate that information because the new strand is the one with the mistake. So your cells have to make sure they can tell the difference between the parent strand and the new strand. So you want to ensure that you have an exact copy of the original double helix. Now, replication is going to begin at a point of origin. And then the replication is going to proceed bidirectionally. And it is always going to go in the 5' to 3' direction. Now, this 5' to 3' direction is going to be from the end to end. So as you add bases, of course, you are going to add 5' to 3', just simply how the ribose is constructed. But you always start at the 5' end of the strand, and you're always going to end at a 3' end of the strand. Now you are going to have a continuous strand and a discontinuous strand. But regardless of which direction it's going, continuous or discontinuous, it's always moving 5' to 3'. Now, your 5' phosphate of the incoming nucleotide is going to bind to the existing 3' hydroxide group. In the DNA chain. Okay. Now, the template strand also you can call that the parent strand, is going to be read from the 3' end towards the 5'. But the elongation, the replication, is always 5' to 3'. Now, in our prokaryotes, it's going to again be initiated at the origin site, and in the prokaryotes, this is called the ORI site, ORI site. And that's the origin of replication. Now, you're going to have a pre-replication complex, which is going to be required to unwind the DNA. So in prokaryotes, they have something called DNA-B, which is their helicase. And the function of a helicase is to unwind the DNA in opposite directions. Now the DNA strands are going to be separated at what they call a fork. So you're going to have your new strands, and they're separated at a fork. And then they're going to be bound with what's called single-stranded DNA binding proteins. It's to protect the DNA. So at the fork, to protect DNA, because now the DNA nucleotides are exposed, and they're exposed to enzymes that could break them down, you have single-stranded DNA binding proteins. And this is going to be the case in eukaryotes as well. And their role is to stabilize the unwound strands. And then you're going to have another protein with respect to our prokaryotes, and this is referred to as DNA gyrase. And DNA gyrase is called DNA topoisomerase 2. And topoisomerases are there to relieve the stress when you unwind. And then DNA polymerase 3 will bind, and its beta subunits will bind to the DNA, and you will start to replicate. Now, just very briefly about DNA, unwinding the DNA. The exact mechanism in eukaryotes and higher eukaryotes is very unclear, but it is absolutely known that the strands of DNA must dissociate and unwind. It's thought that the helicase is going to have a high affinity for one strand of the DNA, and that this pushes the other strand away. Almost like a repulsion. And so that begins the unwinding processes. So helicases are going to unwind. And then you're going to get a region of the DNA that's exposed, and you're going to get these little single-stranded binding proteins to bind to stabilize that now exposed area of single-stranded DNA. These areas are going to now be accessible for the enzyme activities that are going to eventually lead to the replication of the DNA. The replication system with respect to our prokaryotes is going to be our DNA polymerase. And we're also going to have about 20 different proteins and enzymes. In addition, we're going to have these topoisomerases, and these guys relieve stress. And so really the function of a topoisomerase is because you are unwinding here, you are going to be overwound here. And so your topoisomerases have to relieve the stress in that overwound area of the DNA. Now the next thing that has to occur is that you have to have RNA primers in order to extend your, or in order for your DNA to be replicated. And so RNA primers are going to be laid down by the primase activity. And so here we are going to have our RNA primers laid down. And see, for example, our strand is going to be in the 5' to 3' direction. That's our template, and it's the opposite on this end over here. Of course, that's our template strand. And so both strands are going to move in a 5' to 3' direction. So in this case, this is continuous. And on the other side, this over here is discontinuous. And so basically what that means is you're always going to have to go 5' to 3'. So you'll have something here, and then you'll have to stop and you'll have to continue, whereas the continuous side can just very smoothly continue to replicate. It doesn't have any breaks. Now your primers are, of course, important because your DNA polymerase cannot just bind to anything. So your primers will initially lay down RNA primers, and they will be complementary to the DNA, and then eventually they will have to be removed and replaced by DNA nucleotides. Okay. So with respect to E. coli, we are going to have a few polymerases. We have DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. And they're actually numbered just in the way that they were identified. So DNA polymerase 1 is the most abundant. And its major role is to ensure fidelity, and it does this through repair. DNA polymerase 2 is really a backup for DNA replication. It also has the ability to have proofreading, so repair as well. And of course, the main one is DNA polymerase 3. This is the major replicator. It's going to be much less abundant than our polymerase 1, but it's 100 times more active. And so DNA polymerase 3 will have what we call more processivity. And what this is, how long can it remain attached? And replicate. For example, DNA polymerase 2 might only be able to add 1,000 base pairs, whereas DNA polymerase 3 might be able to add 10,000 base pairs at a time. So it stays attached to your DNA molecule longer, and then it's going to be able to replicate a much larger section of the DNA with respect to E. coli. But of course, importantly is the ability to do proofreading. So your polymerase 1 and 2 are going to have a major role in fixing any sort of mistake done by your DNA polymerase 3. So your polymerases in action, you're going to have two central requirements for your polymerase. And the first, of course, is going to be related to fidelity, is you have to make sure that the incoming nucleotides fit. And it must fit the Watson and Crick binding. And then the nucleotide is going to position into the insertion site, and then you're going to have a phosphodiester bond form. And then after this, your polymerase will slide forward and add a new base to the position. And so you're going to have an association and dissociation with your incoming bases, and this is going to affect that rate of your DNA replication. So again, processivity is going to be the average number of nucleotides that are added before the polymerase dissociates. So your polymerase that is replicating must associate plus dissociate. And the number of nucleotides added is processivity. Now the next thing is, of course, going to be fidelity. Fidelity is going to be on the geometry with respect to the base pairs. So base pair geometry. And the hydrogen bonding, of course, with respect to geometry is key to the base pairing. So you're going to have hydrogen bonds between your guanosine and cytidine, which are going to be a fit. If a thiamine came in, you have a lonely hydrogen over here, so that would be an incorrect geometry. Adenosine, thiamine is a correct geometry. Cytosine with adenosine is incorrect. And then also adenosine with guanine is going to be incorrect. So it's really the active site of your DNA pol. And this active site is going to accommodate the base pairs with the correct geometry. So you can think of it as matching. An incorrect nucleotide will bind, but it generally will not fit and will be rejected before you make the phosphodiester bond. Another thing that's associated is proofreading. So this is going to be intrinsic to all of the polymerases. And this is to have a 3' to 5' exonuclease activity. So if the wrong nucleotide is acted, you're going to have the translocation of polymerase to a position where the next nucleotide will be added, and it's going to inhibit the addition until you correct that base that's mismatched. And this is called a kinetic pause. And this is done by polymerase 1 in E. coli. and that's until the mistake is fixed. So again, here you're going to have your wrong base pair added. So here we have a cytosine binding to a thymine. And then you're going to have your exonuclease activity, which is going to occur. And you're going to remove that cytosine base. And then you're just going to add the correct base. So the correct adenine, in this case, will come in and bind. And then you can continue doing replication. And then finally, at the end of all of this, you're going to have ligation. So DNA ligase, you're going to have, again, RNA primers in the liking strand are going to be removed. And then you're going to have, this is going to be done by DNA polymerase 1. And then you're going to have your NICs, which is shown here, going to be filled in by your DNA ligase.